we are going to study about vectors. These vectors are DNA sequences that can transport the gene to be cloned into the host cell. There are several types of vectors are used in genetic engineering. Those are plasmids, bacteriophages, cosmids, phagemids, yeast artificial chromosome, bacterial artificial chromosome, transposons, vectors for cloning genes in plants like Agrobacterium tumefaciens, retroviruses, shuttle vectors. In earlier videos, we discussed about the plasmids. The PBR322, PUC19 we studied in the previous videos. Now, in this video, we are going to study about bacteriophages, cosmids, phagemids, yeast artificial chromosome, bacterial artificial chromosome, transposons, and Agrobacterium tumefaciens like tumor-inducing vectors, and vectors for animal cells like retroviruses and shuttle vectors. The phage vectors. What are these phage vectors? These are the bacteriophages. The abbreviation for the bacteriophage is the phage. Bacteriophages are the viruses that attack the bacteria, or we can say that the viruses that eat bacteria. That is the meaning of bacteriophage. We are using these bacteriophages as a carriers to transport the gene of interest into the host cell. These viruses easily they can infect the bacteria. 1/3 of its genome is non-essential and it can be replaced with a foreign DNA. The bacteriophages have cohesive ends. These are also called cos ends. Cos means cohesive ends. And the non-essential DNA can be replaced with foreign DNA in rDNA technology. You see here, two sites are there for eco R1, and these will cut this DNA into three pieces or three fragments. The middle fragment has non-essential DNA. This is called filler DNA, and this can be replaced with the gene of interest. Thus, rDNA can be produced. Lambda phage vector transports about 23 kb length DNA sequences. M13 phage vector is filamentous and it can infect Escherichia coli. Next, cosmid vectors. These are hybrid vectors, and these are resulted by combining the plasmid with the bacteriophage. In bacteriophage, the cos sites are taken. Rest of the DNA is the plasmid DNA. That plasmid DNA has a ORI site, ampicillin resistant marker, and multiple cloning sites like a normal plasmid. All the essential structures it has. But for that, cohesive ends of the bacteriophages are added. Thus, one genetically engineered hybrid vector is developed. The selection of recombinant transformants is by using antibiotic resistance. These cosmid vectors can carry up to 45 kb DNA inserts. Bacteriophages, they can transport about 23 kb DNA inserts. Next, phagemid vector. These are small plasmids with F1 phage origin of replication, and it has antibiotic resistant site. Say when filamentous bacteriophage origin is there, this vector is smaller than the plasmids. These can transport effectively large sized DNA. Bacterial artificial chromosome. In short, we call them as BAC. It is based on F plasmid of Escherichia coli. F means fertility. Fertility plasmid of the Escherichia coli. It contains the genes for replication and maintenance of F factors, selectable markers, and cloning sites. Look here. This is the ORI site. This is the REP site. It is one antibiotic resistant site, selectable marker, and it has the PAR gene also. The PAR gene ascertains the distribution of large size recombinant plasmids to the next generation. Like PUC19, it also has LAC-Z gene. Many cloning sites also we can find on this bacterial artificial chromosome. So mainly it is based on F plasmid of Escherichia coli, and it can carry very large fragments 100 to 150 kb size of DNA sequences it can transport. Next, yeast artificial chromosome. It contains autonomously replicating sequence of yeast chromosome like ORI, site, etc. And it has centromere and telomeric sequences. It has two selectable markers. This is the marker 1 or X marker. This is the selectable marker Y. And here it has two sites for BAM H1. That is the BAM H1. Bacillus amylopatiens H strain 1. It digests this DNA at this part and forms a linear chromosome with the telomeric ends. Yeast artificial chromosome can carry very large size DNA sequences up to 1000 to 2000 kb size. Tumor inducing vectors. In short, we call them as TI vectors. For example, TI plasmid of Agrobacterium tumefaciens. This is the plasmid of Agrobacterium tumefaciens. It has a tumor inducing DNA. This is called tDNA. By neutralizing the effect of the tDNA, we are using the plasmid to deliver the DNA sequences into the plant cells. Normally, Agrobacterium tumefaciens causes tumors or galls in plants. Transposons. Transposons are short mobile DNA sequences present in the genome. These are a kind of molecular parasites. These DNA sequences can jump or change their position within the genome. These transposons act as vectors for animal cells. Retroviruses. What are retroviruses? These are RNA viruses that can convert the RNA into DNA in the host cell by using a reverse transcriptase enzyme. And such type of viruses are called retroviruses. These are also used as vectors because the viral DNA can become a part of the host DNA. Some vectors are there which can be used for eukaryotic cells and prokaryotic cells. Those vectors are called shuttle vectors. The shuttle vectors contain two types of ORI sites. One is the bacterial ORI and another is the yeast ORI. Example, yeast episomal plasmid and one more is the modified TI plasmid. These can be used for both prokaryotes and eukaryotes.